Hello everyone, it's great to be back to Greece again and see everyone here, those who I know and new as well, that I look forward to come to know. So, and also I would like to thank the organizers, Eleni, Procopius, for the invitation in order to share with you our new findings on the effect of extra virgin olive oil in humans. And this is actually our findings here is from pilot study, which means we are talking about a small number, and we are certainly looking in order to confirm our findings more in larger clinical trials. So our study will be related to studying the effect of olive oil on brain function and memory in subjects with mild cognitive impairments. You can hear me well, everyone? Okay. So this slide is just to show you the difference between, in the left side, how the brain looks in normal people, in normal function. And for the right side is showing the cellular components of the brain that are obtained from Alzheimer patients' brains. So as you can see here, just to summarize what is the important information here is that with Alzheimer patients, you will find that there are several hallmarks or characteristics, including, of course, one of the major one, we know that it is memory loss. And second, we will have the accumulation of what is known as beta amyloid plaques. We have the neurofibrillary tangles. We have a neuroinflammation. We have oxidative stress. In addition, we have that vascular disruption, or what we call the blood-brain barrier disruption. And the blood-brain barrier plays an important role in maintaining a functional brain, because as we age, and more so in Alzheimer patients, you will find that this function is disrupted, and this can result in pathology. So when we started our studies with olive oil, we asked the question that can olive oil and its derived phenolic compounds alter the pathological process of Alzheimer's disease? In other ways, can olive oil and the phenolic compounds like oleocanthal and others presented so far, can they actually prevent or can treat Alzheimer's disease? And in order to answer this question, we started our studies first in animal models. So as you can see here from this slide, we performed many studies in Alzheimer's disease mouse models, and these studies collectively showed that extra virgin olive oil was able to enhance the blood-brain barrier function. It was able to reduce beta amyloid plaques and beta amyloid levels. It enhanced the memory function. It reduced oxidative stress. It reduced the neuroinflammation, as well as it reduced the neurofibrillary tangles, or what we call tau hyperphosphorylation. Now, the question after we confirm it in animals, of course, the objective or the purpose is not the animals. The objective is humans. So the question was, is the EVO effect we observed in mouse models of Alzheimer's disease translated to humans? So in order, which means, in other words, are we going to see the same effect in humans if we give them extra virgin olive oil, similar to that effect we observed in the animals or not? So in order to answer this question, we designed our pilot clinical trial in humans, and we selected subjects or patients. I will not say patients, those are subjects with mild cognitive impairment. Now, while we wanted to study it in mild cognitive impairment, because after all these studies that have been conducted for Alzheimer's therapies, we understand now that the best option for us here is to treat the disease at early stages or even prevent it. So with that being said, so we selected MCI, or mild cognitive impaired participants. And MCI means actually, MCI, it is an early stage before Alzheimer's disease. And usually it happens with age, but it is not a normal process of aging. So with MCI, they have troubles in memorizing things, but still they can go to work, they can do daily life tasks. However, they do not remember everything, they have memory problems, and in most of these who experience MCI, they can proceed further to Alzheimer's disease or other neurological diseases. So that's why we selected the MCI subjects. So how we designed our experiment, remember we are talking about we want to understand the effect on mechanisms. So what we did here, we invited participants to perform MRI in order to monitor the function of the blood-brain barrier. We wanted to see the effect on the brain connectivity. We wanted to see the effect on Alzheimer's disease-related biomarkers. We also wanted to see the effect on memory function, as well as at the end we did also proteomics in addition to other studies, but I didn't put it here because we are still working on the results. So here, just in summary for this slide, this is just to show you the inclusion and exclusion criteria. In order to talk about MCI, we selected those with a specific criteria who are having CDR or what is known as a clinical dementia rating scale of 0.5, and we excluded those who already have some neurological diseases that are unrelated to Alzheimer's disease. So this is the design of our study. So we recruited those subjects, and then we divided them into two groups. One group, we put them on refined olive oil, and the other group, we put them on the governor olive oil. Why we selected the governor here? Because we know that it is rich with oleocanthal as well as rich with other phenolic components. So we used it because it is highly containing phenolic compounds. And then before giving them olive oil, what we did, we asked them to do MRI scan. We conducted memory function tasks, and then we also collected the blood samples and blood samples memory, and yes, so these things. And then after we randomized them, after the baseline, we divided them, and one group received the olive oil, I will call it, and the other one is the extra virgin olive oil. And then we put them on the olive oil for 6 months, and we asked them to daily include the olive oil in their diet, and we gave them about 30 ml per day, or about 2 to 3 tablespoons, and every day. And then we asked them at the end of the 6 months to come back, and we did again repeated the same experiments. We did MRI experiments, we collected the blood samples, and also we did memory function to see how this will affect all these characteristics. So this slide here shows you the baseline demographic and the clinical characteristics. The idea from here that it will show you that those who are in refined olive oil or ROO and those with EVO, actually they were not different between each other with regard to the age, to the education, to the memory function. So they were kind of with same characteristics at baseline. And then those are here, I am showing the results. So you will see in A panel that here it shows the effect on the functional connectivity. What does that mean? We wanted to study the effect of how extra virgin olive oil or refined olive oil can affect the communication between different regions in the brain. So what we find here for the refined olive oil, now just to show you that the green color is after 6 months, the red color is for the baseline levels. So for the refined olive oil or ROO, you can see that there was no significant difference in the brain connectivity, however, between different regions. However, for the extra virgin olive oil, we show that actually the connection and connectivity and the communication between different regions in the brain actually improved. Next, we wanted to see the effect on the blood-brain barrier. And as I told you, with MCI and in Alzheimer's, the blood-brain barrier is becoming leaky. So for this, we used what is known as the DCE MRI. And here we use a contrast. We inject it to the subject. and then we monitor its level in the brain. So we measured that level by the MRI in the brain. So what we found here, while refined olive oil has no effect on the functionality of the blood-brain barrier, measured as permeability, we found that extra virgin olive oil actually enhanced the integrity and enhanced the tightness of the blood-brain barrier as monitored by reduced of the contrast reagent extravasation into the brain. Now this slide shows, just kind of similar to the previous slide, just to show you that how extra virgin olive oil was able to reduce the permeability, and that reduction was statistically different between before and after, and in comparison to the refined olive oil. The results here are actually interesting. So what you see here in under A, here we use two memory tests, CDR, or the clinical dementia rating, for some of boxes, and the other test, which is under B, it is what is known as Whistler memory scale test. So for the A panel, you can find actually those who are in refined olive oil, as well as those on extra virgin olive oil, actually improved memory. As monitored by the green color, you can see it much less than the red color. So obviously 6 months of either ROO or extra virgin olive oil improved memory. In addition, with regard to the other test, you can see here that the y-axis here represent out of the subjects, what is the percentage who has improved in the memory. So as you can see here, for the number of subjects who are improved in their immediate recall, which means remember the information fast, you can see that about 42% of the subjects who were on refined olive oil improved in memory, about 62% improved with extra virgin olive oil. Now for the delayed recall, or LM2, you will find the same here, about 40 and 60% improvement in memory. And for the LM2R, this is for the delayed recognition, which means we come to them after 30 minutes and we ask them to remember the information before 30 minutes. And actually, you can see that about 50% of the subjects in either olive oil or refined olive oil, actually, they had better recall because of the olive oil. So here, for the effect on Alzheimer's disease biomarkers, and here what we measured are the beta amyloid ratio as well as the tau ratio. Beta amyloid is important because of the plaque formation. Tau is important because responsible for the neurofibrillary tangles. So what we found here that either refined olive oil or extra virgin olive oil actually was able to alter significantly the beta amyloid and the tau levels. And in here we can see actually the effect was toward reduction. And while we don't know exactly what is the mechanism behind that, but based on our preclinical studies in animal models, we believe that olive oil, either the phenolic components, as well as we should not forget about oleic acid, everyone. Oleic acid is really important fat in the olive oil that can also have other effects. Now, the phenolic content indeed can affect oxidation, antioxidants, anti-inflammatory, and other things. However, oleic acid also can have some additional beneficial effect. So the idea here from our animal studies, we believe that it affected the clearance as well as the production of beta amyloid that resulted in such effect observed in this slide. Now, for the proteomics, what we did, we collected the blood samples and then we isolated the plasma. We sent them to mass spec lab in order to analyze for us the proteins and whether olive oil altered the proteins in these samples. So when we added all samples together, either refined olive oil or extra virgin olive oil, we found that both olive oils, actually they were able to significantly alter the effect of about 32 proteins in the plasma samples after 6 months of ingestion. And then when we further analyzed these proteins, we found actually that these proteins are play important role in several functions in our bodies, such as it altered the immune system, it altered the coagulation regulation system in our bodies, the antioxidation effect, as well as in addition to other pathways that been altered by the olive oil. While I presented this data, however, we are still refining and working on this, and hopefully we will send it for publication soon. So in order to conclude here, again, I emphasize our study is only 25 subjects, so which means 12 with the refined olive oil and another 13 who are on extra virgin olive oil. So what we can, and more studies are required, so we confirm our results. So what we conclude, at least from our findings from this study, that extra virgin olive oil enhanced the blood-brain barrier function and the functional connectivity between brain areas. And when it comes to refined olive oil, actually it increased the functional brain activation to memory tasks in the brain regions involved in cognition. Both extra virgin olive oil and refined olive oil actually improved memory function in the MCI subjects, and both EVO and refined olive oil altered Alzheimer's disease major biomarkers, which are beta amyloid and tau hyperphosphorylation. Now we all understand that evophenolics, they have important role in the effect that is observed. However, our findings also suggest that the monounsaturated fats such as oleic acid, which is the primary fat in olive oil, could also contribute to the observed effect in our study. And at the end, I can say that olive oil consumption could protect from Alzheimer's disease and prevent or slow down mild cognitive impairment conversion to Alzheimer's disease and other related dementia. So if we ask people to take it as early as possible as they are aging, honestly, I do believe from our study and everyone else's studies that it can delay the conversion of MCI to Alzheimer's disease, or maybe it will slow it down. So of course, this is the acknowledgement slide. We as professors sitting in our offices, it's not us who are doing the job. Those are the graduate students who are doing the work, which are listed under the lab members. And of course, without the collaboration, because again, this is a huge work. It cannot be done by one person. The collaborators, either at Auburn University or at ULM, as well as with Procopius and Eleni here. And of course, the funding from different resources. And I would like to thank the Daphnis family for providing us with the Governor olive oil in order to complete our study. Thank you very much.